Okay, everyone, welcome to the second seminar of the CCSAW Research Seminar series. We've got is it about 30 people online? Yeah. And about 60 people here. Hopefully more will trickle in. I really like getting to triple digits these days. So for those of you who don't know me, I'm Georgia Mason. I'm director of the Campbell Centre for the Study of Animal Welfare. And as this weirdly warm autumn continues, we all agree it's fabulous with a touch of sinister. Let's take a moment to be grateful for the land we're on and to recognise that Canada resides on the traditional unceded and/or territory lands of the First Nations, Inuit and Métis people. And here in Guelph, of course, we're based on the traditional territories of the Mississaugas of the Credit Movement, which is one of the Anishinaabe people. And we're actually on land that was ceded to the British Crown in 1784, and it's under a treaty called the Between the Lakes Treaty, and it covers about 3 million acres between Lakes Erie, Huron, and Ontario. And today that land includes us, but also Hamilton, Waterloo, and St. Catharines. And I know this morning the Brits initiated this treaty because they were looking to create land on which people could settle in loyalists fleeing from the American Revolution. So I thought that was an interesting fact. If you want to learn more about the local history in Mississaugas of the Credit, the Mississaugas themselves have a good website that I'd recommend to you. And then Hamilton Civic Museum has a really great website that's really aimed at families. So it will ask questions like, why should I care? This happened ages ago. And then you can learn about the ongoing disputes around land that was promised and never given. Okay, so without further ado, it's my enormous pleasure to welcome back Emily Miller-Cushon. So Dr. Miller-Cushon is an associate professor of animal behavior and welfare at the University of Florida. And this is in Gainesville, bang in the middle of the state. It's a little island, a little Democrat island in that state. I've actually offered her sanctuary till the election's over. She might take us up on this. Emily is a prolific researcher. She's produced 8 papers already this year, which is stop. You're making us look bad. In 2019, she won the International Society for Applied Ethology New Investigator Award, and she has an H index of 25. Her research program investigates the relationships between management, behavior, and welfare in farm animals. And in particular, she's interested in the development of feeding and social behavior in young dairy calves with the goal of improving their welfare, both when young and in adulthood. So she's a CCSAW alum, and she's actually one of 27 former graduate students who've gone on to create their own research groups and teach and do research in animal behavior. She completed her PhD at the University of Guelph here with Trevor de Vries. What year was that? 2014, after doing physics at Waterloo, actually. So she's a very nimble person. And without further ado, I'm going to hand you over to Emily. Thanks so much. Welcome back.
Thank you so much. It's a real pleasure to be back. It's wonderful. I've been back in Canada in the fall, possibly since 2014. So it's really lovely. And yeah, I'm really glad to be back and to share some of the focuses of research in my lab for the past few years. My seminar talk is titled Considering Social Behavior to Improve Dairy Calf Welfare. And I'll jump around a little bit and highlight some of our research interests for the past years. Okay, so social behavior of dairy calves is, of course, important to consider because they're really unique among gregarious livestock species in that it remains common for them to be housed alone on many farms for the first formative weeks and months of life. So we're going to highlight some research that has focused partly on why we shouldn't do this anymore, why we should be housing calves in social groups, what the benefits of this provide, both short-term and longer term. And this has been a focus of my lab for the past few years doing long-term research to try to understand what effects that early social contact can provide as the pathogen develops and when we enter a stressful social environment later in life. Additionally, as we increasingly move towards social housing, I'm interested in what we can know about how calves that are in social groups. So individual differences in social behavior and group level differences. Yes, I'm sorry. struggling a little bit with this mic. And group level differences in social behavior of calves that are raised in social groups. So this is sort of the scope of what I'll talk about, but I'll start with the first point, this idea of as we move towards housing calves in social groups, why we want to do this, what implications it has for social behavior of the dairy calf, both in the short term and longer term. Okay, so extensive research over the past 20 years or so has shown that there are these benefits of social contact for dairy calves. So social calves from a young age will work for access to a social companion. So they're motivated for social contact. They spend more time near familiar calves. So they develop these social preferences. And then over the long term, they develop bonds for these familiar companions that remain apparent in adulthood. We also generally see reduced fear, avoidance of novelty, and socially housed calves, and all of this aligns with what we know in humans and other species about the negative consequences of social isolation, having long-term detrimental effects on the ability to cope with stressors, resulting in neurological changes and cognitive impairments. So we know this is really important, at least in the short term where the body of research has been done. There are a lot of different ways to house calves in social groups, but really simply just housing a calf with one other friend, pair housing is really common. I think largely this has been looked at in the research setting because it's easy if we think about the need to have experimental replicates at the pen level. And it's also really simple as far as a comparison between management systems where everything remains identical, but for the provision of social contact. So it's nice in some ways, you're controlling everything, but for whether or not the calf has another warm body in the pen with them. So I'll share some both short-term and long-term outcomes from research we've done with calves raised individually or in pairs. So this is for the first 8 weeks of life. They're housed either individually or pairs from within about the first 24 hours of life. They're housed in the same barn, interspersed pairs and individual pens. They're provided the same amount of milk, 8 liters per day. And we've been interested in different short and longer term behavioral outcomes. First, I'll just share some findings that show that, perhaps not surprisingly, consistent with previous work, housing a calf with another calf results in a preference for that familiar penmate. So we looked at this in this study using a social preference test where calves were placed in a previously familiar arena. And we looked specifically at whether the physical contact with the penmate was an important component of forming a social preference for that calf, compared to visual contact with a calf from an adjacent pen. So this is what this looked like. And for pair house calves, the more familiar calf that was presented was their penmate, and the less familiar calf was the calf from the pen adjacent to them. So visual contact only about a pen width distance away. For individually housed calves, they were presented either the calf that was close to them, the adjacent pen, or a calf from elsewhere in the barn, so it's really not familiar at all. And this is a video. There we go. So we looked at latency to approach either calf. We looked at the duration of time with either calf. This is a video that guided students back over the side of the fence so we could have something that's this thing that showed replication. It's a little bit makeshift, but it may do. And oopsie. This is what we found. So not surprisingly, on the left, this is showing that calves raised in pairs spent more time with either calf than custodian. This is consistent with the previous findings suggesting that social housing results in calves that are more comfortable approaching any animal, familiar or not, especially in an unfamiliar testing environment. So they spend more time in close proximity with less than about half body length from that calf, the stimulus calf in the pen. And that was greater than the previous calves or the pair house calves. We also saw that the percentage of time in close proximity that they were near the more familiar calf was greater for the pair house calves. So they preferred to be near their penmate compared to the calf from the pen adjacent to them. Interestingly, individually housed calves had no preference for the calf from the pen next to them to the calf that was completely unfamiliar from elsewhere in the barn. So it really suggests that this physical contact in the same pen is an important component of forming any kind of social relationship. Visual contact, while probably beneficial compared to the previous isolation, didn't seem to actually result in any social preference. Sorry, yes, I'll try not to wander. Okay, so there's also often questions about when calves need to be provided social contact, because even on farms where calves might eventually be introduced to social housing, It remains common for them to be housed individually for the first days or weeks of life. And this is to allow for individual monitoring and control for hygiene. And there's been some work looking at benefits of introducing calves earlier versus later, trade-offs with respect to behavior as well as health. In some recent work, we were interested in what these effects of individual or pair housing for calves that are then introduced to a larger group. So management from birth was identical to the previous study I just described, but then at 2 weeks of life, calves were then mingled between those previously raised in pairs or individually and placed in larger groups on auto feeders. Initially, these were groups of 4 calves, and then they were regrouped into groups of 8 calves. And we looked at some behavioral outcomes, both at the initial introduction to the group pen, and then a little bit later on once they'd adapted to that group environment. So first of all, upon initial grouping, we saw that the calves that had been in pairs from birth spent a lot more time social lying, so lying down within one body length of a pen mate, compared to calves that had been housed individually up until the point of introduction to the group pen. And this was entirely explained by lying near the former pen mate, because at the time of initial grouping, we were looking at putting one pair of calves with some individual housed calves in a group. So they spent more time lying near their pen mate. So consistently, again, they have a preference for their pen mate. It is interesting. So calves spend on average about 16 hours per day lying down. So we can see that the calves that have been raised in pairs were spending a lot of that time near their former pen mate, whereas previously individually housed calves actually seem to prefer to spend time lying down alone. So then moving along, once they'd had a couple of weeks to adapt to the group pen, we did some behavioral tests, and these were outside the home pen, standardized behavioral tests. We did an open field test, we did a novel object test, and these were tests that had been done previously in calves, especially in studies where the testing was done concurrently with the period of differential social housing. So when calves that are in a social environment are tested in these types of tests, generally they approach novel objects more willingly. They appear to be less reactive to being placed in an unfamiliar novel environment. And we were interested in whether any of these behavioral differences in response to a novel environment, response to a novel object, persisted after this period of carryover effects of that early social housing, after this period of adaptation to the group pen. Okay, they did. This is just some results looking at the after subjecting the behavioral data to principal component analysis, the fact that it represented increased duration of novel object contact and shorter latencies to approach the novel object were greater in the previously pair-housed calves. So results that are very consistent with what previous research has shown when calves are actually tested concurrently with being housed in different environments. So there's for at least 2 weeks, which was the time frame we looked at here, there's this carryover effect of early socialization on how they behave during testing in the group pen. And there's some caveats here. In this study, we didn't look at behavior in the home pen at this time point, so we don't know how these responses to these behavioral tests may or may not have translated to differences in their home environment, but it does suggest that there's some persistent differences in how these calves respond to, in this case, this novel environment. Okay, so I've highlighted just a couple of examples of what we know about the short-term effects of housing calves in social environments on the response and their social development, on the response to novelty. Of course, there's other work showing effects on early life growth, on cognition, and all of these effects that we know about in the short term have potential to translate to long-term differences, particularly in ability to cope with management changes, introduction of novel social groups, all the things that we know that dairy heifers will experience as they move into adulthood, and things that are going to have implications for their welfare, as well as their productivity. And there's, of course, work looking at different aspects of socialization across species and how they can have these long-term effects on animals into adulthood. In cattle specifically, there's work showing that prolonged periods of social isolation have effects on longer-term dominance hierarchies, as well as how more complex social interactions like maternal contact can influence social behavior longer term. Relatively, we know less about how typical forms of social isolation for dairy calves of individual housing for just the first 8 weeks of life have longer-term effects on the dairy heifer into adulthood. So this was the focus of some recent work in my lab. We're starting to get some data as calves that had been reared individually or in pairs or in larger groups eventually grow up and enter the lactating herd. So we'll show some examples of our findings from some different points along the way. So first of all, when our research farm, once calves are weaned, so in our case, once they're about 8 weeks of age and having been raised in pairs or individually, they're then in our study mingled between previous housing treatments and put out on pasture around 8 weeks of age. So they go out and it's fairly extensive, natural environment. They're out on pasture. They're given some grain, but they have a lot of space. And so as a first step to seeing if there are longer term differences in social behavior attributed to pre-weaning social housing, we looked at behavior of heifers in their home pen in this pasture environment post-weaning. So we looked at them 10 weeks after post-weaning grouping. So they're about 18 weeks of age at this point. This was a little bit of a challenge from a data collection standpoint. We had a student with binoculars. It was very low-tech live observation out. It was Florida, so we had an umbrella to try to shade him a little bit, but it was a challenge. And they looked at social behavior, looked at social proximity, looked at standing, lying, and feeding, as well as whether or not they were near other animals while doing those things. And as well as just looking generally at time budgets and how animals were spending their day. We worked with a postdoc in my lab, Dr. Katie Burke, who had experience in social network analysis, to look at the proximity-based social networks of the heifers in these groups. So again, these were mingled groups of previously socially housed calves, heifers at this point. And we were constructing the social networks based on the proximity interactions surrounding lying down, as well as feeding. And generally, we didn't see anything, I will say, very exciting. The heifers were pretty similar. I think one of our major takeaways was that the present social context in this environment might have been more influential than previous social experience, because we did see some differences between groups. But one thing we did see was that the pre-weaning pair assignment explained the structure of the social network. So in other words, we saw that the calves from the same pre-weaning pair were more likely to be spending time near each other than other pairs of calves that hadn't been housed together. So this graph shows that just the network from one group, and so the circles represent individual heifers, color-coded based on pre-weaning housing treatment. The thickness of the line represents the frequency of proximity interactions between those pairs. And so one of the first things we looked at was the centrality. So in other words, how connected was a single heifer? How many interactions did she have with other heifers within the group? And we didn't really see any major differences attributed, like I said, just to pre-weaning housing treatment. But the blue lines on the right-hand side of this graph show those heifers that were in the same pair pre-weaning. And like I said, they were more likely to be interacting, spending more time together than other pairs of calves. So again, consistent with some of our short-term behavioral tests suggesting that the heifers develop a preference for their pen mate. And in this case, it seems to persist, at least in some cases, up till this 10 weeks after they have been housed with just that single pen. And I should say too, that for the most part, these groups were not completely stable over this 10-week period, but largely pen mates were kept together. They were moved through pasture pens as they aged. So there was some introduction of new animals here and there, some consolidating of smaller groups as they got older. But as you can tell, based on the fact that these calves were still with their pen mate, there was some consistency in the group. Okay, so moving along, at our farm, about 30 days before expected calving, the heifers were introduced to a freestall pen for the first time. And this is a pretty stressful, I would say, event. It's a new social group. At this point, they're largely without their former pen mate because it kind of depends on the timing of when they're going to have. So the first time they're seeing a freestall, so it's a novel physical environment. They have to figure out where to actually go and where to lie down. New animals. Our farm manages mixed parity groups, so it's not just heifers calving for the first time. There's bigger, older animals in the group. Stocking density is variable, as I'll mention again in a moment. So it's a novel transition, social transition, housing transition. So we looked at their behavior 24 hours following this introduction to the freestall pen. And we were interested in partly how they were using the space. So were they standing, were they walking, how were they using the stalls, how much time did they spend eating, as well again as their social interactions. So were they in the stall next to somebody, were they feeding next to somebody, displacements from the feed bunk, displacements from the stall. And here we found some more dramatic differences between previously individually and pair housed calves, which initially surprised us because it was this later time point. We hadn't seen anything really dramatic on pasture. But I think it makes sense when we think about the fact that we had thrown them into this much more stressful environment where there are going to be differences in coping ability to cope with these transitions. This is where it's going to come out. So just very briefly, one of the major things we saw was that previously pair-housed calves spent more time eating, they spent more time walking, but not more time lying down. So when they were standing up, they were just moving around more. And they spent less time in the stall. But maybe most interestingly, so we had this sort of natural fluctuation of stocking density. This is something outside of our control. So we just characterized it at the time of observation based on categories of stocking density. So we had low stocking density, where there were less than 75% of the stalls were accounted for by a heifer in the pen, medium stocking density, and then high stocking density, where there are more heifers than stalls. And for some of our outcomes, we had this interaction between stocking density and previous housing treatment, where differences became most apparent at high stocking density. So this is shown, I think for feeding, you can see, so overall there was this effect where previously pair-housed calves spent more time eating, but it was most apparent at high stocking density, where individually housed calves kind of dropped off more and pair-housed calves were relatively less affected. And then also for stall visits, which I thought was really interesting. We didn't have any particular predictions about how stall use might differ, but pretty quickly it was one of the biggest things we saw affected by pre-weaning social housing, where particularly at high stocking density, we saw that previously individually housed heifers seem to respond to their patterns of stall use seem to respond to increasing stocking density in a way consistent with other access to resources in the presence of competition, where stall use, their visits became less frequent, but the duration became greater. And it seemed that they were perceiving maybe a greater barrier trying to access the stall, and also that their use of the stall was more consistent across all stocking densities. So suggesting that they were maybe, even though they weren't lying down more, they're maybe viewing the stall as maybe a safe place to be. They're more likely to be spending time there as opposed to walking around, exploring the environment or feeding. And so yeah, we saw this with respect to the duration of stall visits. And we also saw that overall, regardless of stocking density, that previously individually housed heifers were more likely to be displaced from a stall. This might have just been because they were in the stall more, but also suggests that maybe they were experiencing some increased competitive pressure. Okay, so I'll jump a little bit. So we have, this evidence, of course, of short-term benefits of social housing. We're starting to see some interesting things with longer-term effects. This is an area we're continuing to work on in my lab. But we still see on farm that a fraction of producers are housing calves individually, and some recent work we were interested in how dairy producers actually perceive social housing, some of the barriers to it, as well as their perception of how it has implications for both aspects of the calf's behavior and maybe their welfare in that environment, as well as whether they have any concern about calf health and in social housing, which has often been described anecdotally as a concern surrounding social housing, even though research generally suggests that there is not much of a risk associated with at least smaller groups of socially housed calves with respect to health. So this was a survey study we did last year, a couple years ago. We had about 100 respondents across different states in the United States, and we had largely targeting calf managers and owners of farms. And we asked them questions about how they perceived social housing to have implications for calf comfort and calf health and performance. We chose calf comfort as kind of a proxy term based on previous surveys suggesting that producers use this term, and it seemed to sort of related to aspects of the subjective emotional experience, trying to get at kind of how they think the calf feels in different housing environments. And generally, we saw this result where respondents appeared to think that social housing was pretty good for both calf comfort, and slightly more ambiguous results with respect to calf health, where there was a little bit of a response rate suggesting that some producers did have a concern about health associated with social housing, but generally skewing positive for both. And also interestingly, we saw that of the respondents that were already using social housing on their farm, they were more likely to have an even more positive perception of social housing for both calf comfort and calf health. And I think this could explain one of two things. One is it's entirely possible we had a large percentage of our respondents who were decision makers on the farm who had a pre-existing positive perception of social housing, and that's why they were already doing it. But also I think it shows that if they were seeing it, if they had practice actually implementing on the farm, it may have improved their perception of it, which I think is a nice finding. Okay, so regardless of the benefits that I think are pretty conclusive with respect to social housing, we're moving there one way or another, especially I guess in Canada by maybe 2028. Elsewhere, hard to say, but we're moving that direction. So I've become kind of more interested in what we can actually know about calves in social pens. So what are they doing, especially in larger groups, which have received relatively less study to date, because I think probably because of some challenges associated with replicating experimental units, and we'll talk about those a little bit more. But on farm, especially large farms, which we have more of in the US, feeding calves by auto feeder, it can be common to have really big groups of calves. And I wonder a little bit about individual variability, group level variability, what changes in social behavior over time can tell us. So I'm going to talk a little bit about what we've been asking about, more questions than answers at this point, I think, but interesting to think about. So going back a couple of years, one of the first things we were interested in was just individual variability in social behavior. So it's quite a bit of work suggesting that both that there are consistent individual differences in social behavior and differences at different time periods and over time, respect to different states. So of course, things like social withdrawal surrounding parturition we see in livestock species, work across species, including humans, showing changes in social behavior in response to pain. So humans experiencing chronic pain and social isolation, depression and social isolation, work across species, looking at changes in social behavior related to health. Interesting work in wildlife disease ecology, showing changes in patterns of social interaction associated with disease, and even suggestion that's evolutionary mechanism to reduce disease transfer, where healthy individuals will avoid sick individuals, and sick individuals will maybe avoid social interaction to conserve energy. And then, of course, consistent individual differences, which can be attributed to personality, including in dairy calves, where there's evidence that differences in selectivity of social partners can relate to pessimism, relate to other personality traits as well, suggesting that we could be trying to dig more into both stable individual differences across time, what they tell us about that individual, and then within individual changes that might tell us something about that individual's welfare. So initially, I was interested in this health component, this idea, health, disease is a challenge in dairy calves. We understand more about their social behavior as maybe an indicator of health status. So a few years ago, I took advantage of a disease challenge trial where there was calves were inoculated with Hymenia hemolytica. It was a short-term response. They developed a fever and then back to normal the next day. And this was nice to have a relatively small number of calves in a fairly controlled setting. We had groups of 6. We challenged half of them. And we looked at changes in behavior. So the graph here is showing reductions in social lying bouts. We also saw reduced social grooming in the calves that were challenged on the day of the challenge and then back to normal the next day. We also dug into this data and looked at actual social contacts directed and, initiated and received. So we looked at both social grooming and also just kind of simplified a little bit. Anytime a calf went up and touched another calf with its nose or received contact from another calf. So sort of the directed active social contacts, as well as undirected, more passive social behavior. So social lying next to another. And so with these different data sets, we again constructed social networks, both directed ones that took into account the initiated and received interactions and undirected ones. And consistently across all of this, we saw pretty dramatic differences where the sick calves had just less social interaction. So they were pretty lethargic. They were initiating less social contact. They were receiving less social contact. They were more likely to be lying by themselves, which I also thought was interesting. Maybe not surprising that they're not actually initiating active social interactions if they're not feeling well, but they're also more likely to be lying alone. So again, sort of suggesting this, at least in this disease challenge model, changes in social behavior associated with health. But of course, there's some limitations here. We don't necessarily know how well this maps to the real world. calves developing disease naturally. But the things that were nice about that study were that it was small and it was predictable, and we knew when they were going to get sick and how we could watch the video. So we took a different approach to try to answer this question in more of a realistic setting. So here we looked at clinical health, bovine respiratory disease, using scoring throughout the week for temperature, for coughing, for nasal discharge. But this would not have been practical to look at using video analysis like the previous study. So we looked at an ultra-wideband location tracking system. This is the NOLDIS track lab system. And there's some limitations here. We characterized here proximity-based social networks based on a threshold of 1 meter. We lose some nuance here. We don't really know what they're doing. We could dig into it more and combine it with activity data to know if they're standing down or lying up. Have not done that yet, but it's at least some measure of how much time they spend near other calves. And in some ongoing work, we are trying to relate the actual positional data we get from this, the proximity-based social networks, with sort of some focal calves and days for video analysis to look at like active grooming or actual social behavior to see how it actually relates to the proximity-based networks to get a better sense of what we're actually measuring when we look at this data. But because that takes a long time, we jumped into looking at how these proximity-based social networks align with clinical health status and subclinical health status, I should mention. So we also, in addition to the clinical health scoring, had lung ultrasonography to look at lung lesions as an indicator of subclinical respiratory disease. Okay, so this is one thing we found where individual lung consolidation during the weaning period was associated with reduced centrality in the social networks. This is shown for 3 groups. We had 9 groups of 10 calves per group. And the calves shown in gray are those that had lung consolidation. And this is during the weaning period. So what we saw was that calves with lung consolidation during the weaning period were strongly predicted by prolonged periods of clinical respiratory disease during the pre-weaning period. So essentially what we're capturing here is those calves that had a rough go of it, were sick for a long time, showed up during the weaning period as less central in their social network, which I think is maybe not surprising. We didn't see during the pre-weaning period that individual health status predicted any aspect of centrality in the social network. But there's a caveat to this I'll come back to in a moment. First, I want to jump to this, which looks a little bit complicated. So because we had so much data from this location tracking system, we knew all the calves all the time, what they were doing. We wanted to more generally understand what the social networks looked like over time between individuals across life stages. So these graphs are showing for all 9 groups, strength centrality, which is the line over time. So these are the weeks of age, so weeks of data collection, and pre-weaning, weaning, and post-weaning are shown with the different colors. So for any of these groups, we can kind of look at them. We saw that to some extent centrality measures were repeatable across weeks. So there was some consistency. The calves, if you look at any single line, the calves that had the highest strength centrality, so in other words, the most connections, most interactions with the calves in the group, during the first weeks of life, were generally speaking more likely to be those that had the most connections with other calves in the group later in the mating period. So some evidence of individual consistency. We also saw that on the whole, the amount of interaction within the group was affected by rearing stage. On average, we saw that calves, their strength centrality decreased during weaning. But big caveat here is that there was so much variability between groups. So the variability, if you look at the scale on the y-axis, there's actually variability in how much interaction any one group had. Some groups just interacted less than others. And there's variability about how the groups responded to weaning. So some groups, centrality actually went up. They were having more interactions within their group during weaning. In some cases, it went down. And this was, we don't know what's going on, right? It's like there's so much variability here. And there was a little bit of variability in composition of the group. So all groups had heifers and bulls. Some groups had a few more heifers and bulls, a little bit of an age range, but they were all within 10 days when they were grouped. The groups were stable. They're all Holstein calves. They were all enrolled around the same time period. So, the factors explaining these group level differences in social behavior, not very well understood. And this was really interesting to think about, I think, from as far as methodological considerations for future research, because we had a lot of calves on this trial, and that's not always feasible. So when I think about when we're looking at collecting data from a few groups of calves, I think it's hard to know how many groups we really need to have data describing social behavior with any degree of external validity. Because if we'd only enrolled a few of these groups, we would have not necessarily seen that how social networks respond to weaning is apparently quite variable. So with that, I'll come back to our health results with another confusing graph. And this was showing that individual health status was not the most influential thing when looking at social network centrality. We saw that the group level disease prevalence was by far the strongest predictor of strength centrality, which I think makes a little bit of sense if we think about how we're probably going to be doing less socially if all of our friends are sick. This again is showing it for all 9 groups, and the 2 box plots side by side for each group present different weeks of data collection during the pre-weaning period, weeks 3 and 5 of age. And the box plots are color-coded to show at each time point the prevalence of clinical respiratory disease. So the more red boxes are the more sick ones, and blue ones are the ones that are healthier. So generally, again, there's some variability, but generally when the group had a greater prevalence of disease, the number of interactions within the group dropped. Of course, there's variability here, but on the whole, this is what we saw. And yeah, this completely swamped any, there were no indications that individual health status was going to predict centrality within their individual group. It's entirely, in this data set, dependent on how many of their calves were sick. And this is consistent with some other work in calves showing differences in social play behavior based on the health of the companions, which again, it makes a lot of sense when you think about opportunities for social interaction. You're going to depend on what else is going on within your group. And of course, again, we don't necessarily know what explains the variability between groups. It could be something as simple as, who in the group gets sick is going to make a difference, because it probably depends on the relative position of any of those calves, how social they were. If a very social calf within the group gets sick, that's probably going to affect the group disproportionately. And that's just nuance we don't really have any insight into at this point. So I think this, again, raises more questions than it answers maybe. We were interested in sort of in these group level differences. Of course, it makes sense. Social behavior, it's not about individuals necessarily, it's about their social context. It's what's going on in your group. So a couple more things we've been thinking about. One, I'll show an example, just thinking about what group level factors might influence social behavior. So this is from a study where we looked at space allowance for groups of calves. And this is a heat map showing where calves are in their pen at different space allowances. So this is for just a couple different example pens. On the left-hand side, these are calves that had 3.7 meters squared per calf. And we can see we achieved the smaller space allowance just by kind of blocking off part of the pen. And this is comparable to sort of minimum recommendations for group house calves, and then increasing to 4.6 meters squared per calf, and then up to 5.6 meters squared per calf. So generally a little bit higher than what we'd be expecting to see on the farm today. So at sort of at this group level, this heat map using computer vision shows that when calves have more space, they seem to like to be near the edge of the pen. We have a little preference for the perimeter near the fence line, which is something that's been seen in other studies as well. And then as space is reduced, essentially they go everywhere because maybe they don't have the same ability to choose sort of periphery areas. So in this study, we also looked at activity, and we looked at the synchrony of activity. And we saw that increasing or decreasing the space allowance didn't affect line time, but increasing space reduced the coefficient of variation in line time. So again, no difference in average pen level line time, but when you look at individuals, some were maybe increasing line time, some were maybe decreasing line time. They're kind of disproportionately affected. But on the whole, it was not really this actual average trend either direction. Number of lying bouts increased, and when we looked at the diurnal patterns of activity, the synchrony, or the hourly interclass correlation, actually increased as well. So we've got them more synchronous in their activity when they have more space, and this maybe might be due to the fact that they can kind of have the preferred resting spaces. We maybe expect them to prefer to do the same things, and they're more likely to be able to do the same things when we have a little bit more space to accommodate them. So I think it gets a little bit at maybe what individual differences there might be within the group, differences in Could be differences in preference for social contact with other calves. Maybe it's easier for them to also lie down if they're at a preferred distance from their pen mates. And generally, maybe what else we need to do to better understand at the group level what socially housed calves need to accommodate preferred patterns of social behavior. So, slightly more off the wall example of this from a couple of years ago. We were interested in what socially housed calves might do if they have the opportunity to be not social, sort of. So this was a shelter, a three-sided shelter that kind of just gave them a little chance to escape from the group with a little bit of visual isolation. We weren't actually stopping other calves from going in there with the calf. Turns out they did this quite a lot, which we hadn't really expected. But, you know, of course, there's nuance in what we all want from social interaction. Like we can all agree unanimously that social contact for calves is important, but at the same time, there might be moments where they need to be alone or they want to avoid, you know, more rambunctious pen mates. And so we looked at this sort of shelter design, and initially we saw huge variability in calves using this. So some calves were in there for 20 hours a day. That's more time than they spent lying down. They wanted to live in there. Some calves maybe went in there for an hour or less. They would check it out and they would leave. And so for this study specifically, we were interested in the shelter use surrounding disbudding. So again, disbudding is painful even when we give local anesthetic and analgesic, which we did. But yeah, work across species suggests that when individuals are in pain, they might reduce their social interactions. And so I guess we predicted two things. One is that disbudding, which is painful, would motivate them to seek out social isolation. And then also that this shelter would accommodate that, which it may not have, but we'll get to that in a sec. So we did see that in group housed calves, those that were disbudded, increased use of the shelter compared to their pen mates that were not disbudded, particularly on the day following disbudding, which is probably when the analgesic was no longer effective. So it did seem that they wanted to hang out there when they're experiencing pain following disbudding. But we also did see, like I said, that a lot of them are using the shelter together. So whether or not they're actually using it because it accommodated their preference for social isolation is hard to say. We did see that the disbudded calves compared to their pen mates were more likely to enter the shelter when it was empty and then leave it when there was another calf there. So maybe they were trying to use it for social isolation, but wasn't always accommodating that. And we did repeat this study in pens with, where calves were provided two shelters to see if we could kind of give them a better shot at actually being alone if they wanted to be. And we haven't finished working with that data yet, but I think just generally it's interesting to think about how we can provide different aspects of environmental complexity, even just increased space or more opportunities for sort of visual barriers. Calves like to maybe be near the edge of the pen, you know, for easier vigilance behavior, and how that complexity can accommodate individual differences, either consistent individual differences between individuals in terms of what they want in terms of social interaction, or within individual differences when they are experiencing pain or when they are getting sick. And of course, there's this ongoing challenge in how to actually characterize this behavior, anything to do with pen use, social interaction. I think computer vision is really exciting because in addition to just where the calves are in the pen, we can look into a little bit more nuance as far as how they're actually interacting, something that currently with the system I'm using, with the location tracking system, we don't know anything about. Having spent hours and hours of my life watching video at different points and also subjecting my graduate students watching video, it's exciting to see what scope of data we might be able to collect, with the help of other tools. And I think as we learn more and more about individual differences and group level differences, and we see the need to actually understand the behavior of calves in these larger groups that are representative of on-farm conditions, using other tools like this, I think is exciting. Okay, so I guess I'll wrap up with some questions. I think some not beyond the scope of what I'm able to answer. I'm trying to answer right now, but things I think we should be thinking about. So one, I think, what is enough to meet a calf's social needs? And is it really just putting them in a pen with another calf the same age, which is sort of where we're heading now in the dairy industry? Do we need to consider maternal contact? There's certainly conversations about cow-calf contact, even within pens of roughly same age calves. What else can we provide to them to accommodate preferred patterns of social behavior? Should we consider, more age heterogeneous groups, or other opportunities to vary in how social they are within the group? And then these questions of long-term effects of really social experience, and I'm thinking not just long-term effects of social housing compared to individual housing, but also long-term effects of different types of really social experience. So housing calves with one other calf in a pair or larger, more complex groups, effects of group dynamics, how all these things might be more likely to set up a heifer for longer term success, more likely to help her develop social behaviors that improve welfare long term. And then finally, what we can learn about individual and group level differences. I think some of this is important to be able to refine management, maybe accommodate differences in social groups, differences in individuals, but then also what we can learn from this as far as assessing welfare. So changes in social behavior that may predict health or pain or positive affective states to try to use it as an indication of welfare with the help of technology to make all of this data easier to collect. So with that, thank you very much. Happy to take any questions.